Glycolysis is an anaerobic process used to break down a glucose molecule into two molecules of ATP and two molecules of pyruvate, which are better able to be used by organisms in various ways. Pyruvate can either be used in fermentation in the absence of oxygen to make lactate or ethanol, or it can undergo oxidation and enter into the citrus acid cycle to make even more molecules of ATP. Organisms store energy as glucose because it is the most stable of hexose, but it is difficult for organisms to use. Glycolysis contains many steps to create pyruvate from glucose, so the activation energy for any individual step is not so great that it takes more energy to perform glycolysis than is produced. Glycolysis has two stages. Stage 1 involves trapping glucose in a cell and cleaving it into two phosphorylated 3-carbon compounds. This stage invests ATP in order to cleave the molecule. The second stage is energy harvesting, and it involves oxidizing these compounds to pyruvate and to generate two molecules of ATP.
So there's three regulated steps in glycolysis, and one of the three is the very first step, which is the addition of a phosphate group to glucose, making it the glucose 6-phosphate by hexokinase. This step is a group transfer reaction that allows or involves the cleavage of a phosphate group from one of the two ATP molecules input to glycolysis to allow it to function. The cleaved phosphate is then added to that glucose molecule by the hexokinase. And this is regulated by its own product, the glucose 6-phosphate. So the G6P allosterically binds with the hexokinase, which means not at its active site, and inhibits it from allowing the transfer of the phosphate group from the ATP to the new glucose molecules. So this means that if there's too much glucose 6-phosphate in a system, its own overproduction is going to inhibit further overproduction, essentially keeping the level of energy both from the ATP produced at the end of glycolysis and the pyruvate at the end of glycolysis to be converted to more ATP in check. And I have a diagram here just to show basically what I'm trying to say. There. So the glucose is converted via hexokinase to the glucose 6-phosphate. That's the phosphate group that's been added. It's just P, but it's a phosphate group. And then the glucose 6-phosphate goes back and allosterically binds to the hexokinase, inhibiting it from further overproduction of glucose 6-phosphate from the glucose.
And with regulated steps comes unregulated steps. So two unregulated steps that I picked was the first one was a reaction catalyzed by triose phosphate isomerase. This is sort of a side reaction to glycolysis. This reaction turns dihydroxyacetone, another product of aldolase, into the better, more hip cousin molecule, glyceraldehyde 3-phosphate. This is important because when the ketone present in dihydroxyacetone phosphate turns into an aldehyde by the triose phosphate isomerase, it gives way for the aldehyde to be easily turned into 1,3-biphosphoglycerate and later on into an acid catalyzed group. Without this important step of the creation of glyceraldehyde 3-phosphate, we wouldn't be able to create the high phosphoryl transfer potential that we see with 1,3-biphosphoglycerate. And then following that is pretty much the step almost right after that, is the transfer of the phosphate from 1,3-biphosphoglycerate to ADP to make ATP. This step, while not the most flashy of steps, is one of the most important ones, as it's one of the two steps in glycolysis that actually makes ATP for a body to use. Usually ATP and ADP are the ones that hydrolyze their phosphate groups to make reactions exergonic. However, 1,3-biphosphoglycerate is so thermodynamically unstable, it is able to transfer one of its phosphate groups to ADP to generate ATP. And finally, this reaction is catalyzed by the enzyme phosphoglycerate kinase.